Hi everyone. So this is the toughest questions that came in INICET explained in a very short time, which we are covering a few questions very frequently asked by the students. So first question is, this is a graphical representation of a titration curve of a biochemical compound. Which of the following statement is true? So before coming to the answer, let me explain what is a titration curve in a very short way. So first and foremost, understand that titration curve is a graphical representation of two things. That is the pH of the medium against the alkali being added. Right. So if this medium is having a compound with a single ionizable group, for example acetic acid, it has one ionizable group. So if it has two forms, one is the unionized form and the other is the ionized form, where the ionizable group is getting ionized, right? So the first is when we start adding alkali, this is the point, when we start to add the alkali, the unionized form, which is called as CH3COOH, will be more, but at the midpoint what happens is the number of ionized is equal to the unionized, which means the form 1 is equal to the form 2, which is the ionized and the unionized form. This is called as the partially ionized state. And the last what we can see is all of the acetic acid will be completely ionized. So form 2 will be more. So ionized is more than the unionized form. So these are the 3 points. Now you see the changes in the pH when we start to add the alkali. So initially when we start to add the alkali, what we can see is the pH is varying a lot. But later what we can see is that the pH is almost remaining a constant like this. So the change in the pH is only this much. So and the amount of alkali that you have added is almost this much, which means that even if you are adding the alkali, the pH of the medium is remaining constant. But after that what will happen, once the ionizable group is completely ionized, then what happens, the pH is changing a lot, right? The pH is changing. Even a slight change in the alkali will markedly affect the pH. So this is what happens, which means that in a solution, the solution is having the maximum buffering capacity is at the point where the component or the compound that is present in the solution is in a partially ionized state. This is called as pH is equal to pKa. And what is the buffering range? The pKa plus or minus 1. So the buffering range is pKa plus or minus 1. This is the range where it is having maximum buffering capacity. So it means that the midpoint is the point at which it is called as the ionization constant, otherwise pKa. So this, when pH is equal to pKa, it is having the maximum buffering capacity. Now you can see that this is a compound with a single ionizable group, that is acetic acid. Now you see this picture. In this picture you can see a titration curve itself, but this is having two curves. So this is the first and you can see this is the second titration curve, which means that this is a compound with a two ionizable group. For example, amino acid, it has an amino group and an acidic COOH group, which means that there are two ionizable group and there are two titration curve. So the midpoint of the first titration curve is the ionization constant of the first ionizable group, and the midpoint of the second, so from this onwards it is the next titration curve, and the midpoint is the ionization constant of the second ionizable group. Okay. Now if you take the midpoint of these two, that is the average of pK1 and pK2, that is what is called as the isoelectric point, otherwise Pi. Now you see the variation in the pH. When the pH is equal to pK1, then it is having maximum buffering capacity. The pH is not varying. Now when the pH is equal to pK2 also, the pH is not varying much. But you see the point at which pH is equal to Pi. When the pH is equal to Pi, you can see the pH is varying a lot. The pH of the medium is changing even with a slight change in the alkali. So which means that at pH is equal to pK1 or pK2, then it is having maximum buffering, or partially ionized state, it is having maximum buffering capacity. But when pH is equal to Pi, it is having least buffering capacity. Now you see this picture. Tell me how many ionizable groups are there, seeing the number of titration curves. So this is one titration curve, again this is another titration curve, and this is the third titration curve, which means that this is a compound with a 3 ionizable group. For example, aspartic acid, it has an extra carboxylic acid group. So like that, if it is having a 3 ionizable group, then you can see 3 titration curve. Now we come back to this question. So in this question, what you can see is the maximum buffering capacity is represented by point A and B, which means this is a titration curve with the compound having 2 ionizable groups. So this is the first titration curve and this is the second titration curve. So there are 2 points here. One is, this is the pH is equal to pK1. This is pH is equal to pK2, point A and point B. So the maximum buffering capacity is represented by A and B. Is it right? Yes, that is correct. Now, point A and B represent the maximum ionization. No, maximum ionization of any particular ionizable group is the end point of that particular titration curve. Like that, this is the maximum ionization, that is the end point of the first titration curve, and this is the maximum ionization, that is the end point of the second titration curve. Then this compound has 3 ionizable group. Is it right? No, because it is only 2 titration curves, so it is definitely 2 ionizable group. It's a wrong statement. So maximum ionization at point A and B, that is also a wrong statement. This compound has 1 ionizable group. No, it has it has 2 ionizable group, right? Because we can see 2 titration curve, which means the answer here is A. So I hope that is clear to you, and this is what you have to learn in the topic of titration curve. The next question we are discussing is about electron transport chain inhibitors. So this is actually a experiment which is we are conducting on mitochondrial respiration. You add malate and pyruvate, which means it is having a substrate to donate the electron. The respiration is normal. You add succinate, again it is another donor of a electron group, so which means again the respiration is normal. In the presence of pyruvate and succinate and malate, in spite of all these presence of these compounds, something else is being added and respiration is blocked. Which one of the following is most likely added? So to understand this, let me tell you what are the effects of various inhibitors of electron transport chain. So just see this picture, this is the electron transport chain which is located in the inner mitochondrial matrix. This is complex 1, this is complex 2, this is complex 3, and this is complex 4, and this is the ATP synthase complex. Okay, so the inhibitors are of different varieties. Number one can be an inhibitor in the electron transfer, which means any inhibitors from complex 1 to complex 4, these are the inhibitors of electron transfer, and there are inhibitors of oxidative phosphorylation, and there are something which is called as the uncouplers, which uncouples the oxidation from the phosphorylation, right? So now if the blocking is in the electron transfer, what happens is if the blocking is late, which means that complex 4 is blocked, what you can see as an effect is the oxygen is not at all getting any electrons and oxygen is not reduced to water. This is what happens. So if the block is in any complex, then oxygen, this is called as consumption of oxygen or respiration. If the block is in the electron transfer, then oxygen consumption is reduced, respiration is blocked. But there is one more point here. If the blocking is late, that means if the blocking is at cyanide, then the electron transfer is suddenly stopped and oxygen consumption suddenly stop, respiration stop, and hence ATP production also stop. But if the inhibitor is before that, which means is it in complex 1, if it is in complex 2 inhibitor or a complex 3 inhibitor, then the electron transfer is not completely blocked. The complete blocking and sudden stopping in respiration happens if it is cyanide. Before that anything is added, that stopping is not so sudden. This is one thing that we should keep in mind. Okay. Second is inhibitors of oxidative phosphorylation. Inhibitors of oxidative phosphorylation, for example, oligomycin, venturecidin, they all act at the FO subcomplex. So, FO subcomplex, we know that it is a proton channel where the protons are being transferred. So, if that proton transfer is being blocked, what happens is the oxygen, the ATP production will suddenly stop because of this H plus transfer, ADP is getting converted to ATP. That is first thing, ATP production stop. Then what about the oxidation or the respiration? So in Leninger, what is given is that if the blocking is in the oxidative phosphorylation, then the two things will happen. ATP production stops and oxygen consumption is also stopping. Oxygen consumption will also stop because these are complementing each other. Oxidation is coupled with the phosphorylation. So, if phosphorylation is not happening, oxidation is not also happening. So as a result, what happens is oxygen consumption will be stopped and ATP production is also stopped. That is the effect of inhibitor of oxidative phosphorylation. The first thing is ATP production stop, and because of that oxygen consumption, because for everything we know that ATP is required. So if ATP production is stopped, so naturally the oxygen consumption is also going to stop. So that is the effect of inhibitor of oxidative phosphorylation. Then the uncoupler. Uncoupler means if you add an uncoupler, oxidation is uncoupled with phosphorylation, which means that oxidation will happen but phosphorylation is not happening because they are not coupled with each other now. So oxidation will happen and oxygen consumption is happening, respiration will happen, that means oxygen consumption is happening, ATP production will definitely stop because we know that as oxidation is like that uncoupling happens, the proton gradient is not there, which means that these transfer of proton through this FO subcomplex is not taking place, which means that ATP production will stop. So these three effects you should be thorough with. So if the block is in an electron transfer, then oxygen consumption is stopping, ATP production is stopping. If the block is in oxidative phosphorylation, ATP production definitely stop, oxygen consumption is also later stopping. Next, if the inhibitor is an uncoupler, then oxidation will take place. In fact, it increases ATP production stop. Okay, so I will explain it with this picture, a very simple way. So what you can see is, this is a picture given in Leninger. What you can see is that if there is succinate and a source of ADP is there, then what you can see is, you can see here the oxygen consumption and ATP production is actually taking place. So the red line is oxygen ATP production and the black line is this oxygen consumption. So both are taking place. Now when you add venturecidin or oligomycin, it is an inhibitor of oxidative phosphorylation. What you can see is ATP production suddenly stopped. Now what about oxygen consumption? Oxygen consumption initially is not stopping, but later what you can see is that it is also reducing, it is not increasing. See what increases this curve like this. So there should be an increase and a sudden rise, or there should be an ascent in the curve. It is not happening. So oxygen consumption is also stopping. ATP production is also stopping. Now what you can see, a dinitrophenol is added. Dinitrophenol is an uncoupler, which means ATP production is stopping, oxygen consumption is actually happening here. See, you can see here there is a increase in oxygen consumption. So uncoupler, oxygen consumption increase, whichever others are there, oxygen consumption stop, ATP production stop. This is what we should understand. Now again you can see here, this is another picture where you can see there is ADP, there is a source of electron and cyanide is added. Cyanide means it is a complex 4 inhibitor. So complex 4 inhibitor means that suddenly the oxygen consumption will stop, ATP production is also stopping. Now we come to this question. So which is the most likely compound that is being added? That is a question here. Rotenone, antimycin A. These two are inhibitors of electron transfer. Oligomycin is an inhibitor of oxidative phosphorylation. Dinitrophenol means it is an uncoupler. So the question here is, which is the one which stops the respiration? We know that the respiration is increased by dinitrophenol or an uncoupler. So this is out of question. Now we have to select between these three options, which is the one that stops the respiration. Now, so in this, oligomycin is an inhibitor of oxidative phosphorylation. Rotenone, what is it? It is a complex 1 inhibitor. What about antimycin A? It is a complex 3 inhibitor. Now you see this picture. If rotenone is added, then so whatever complexes or redox couples prior to that, it is not oxidized, but you can see here. See the electron donor, whatever prior it becomes reduced, and whatever after the block, that is what is given here, after the block become oxidized. Okay, so if it is rotenone, what you can see is this is reduced, but rest everything is getting oxidized. If it is an antimycin A, whichever redox couples are prior, it is getting reduced, but this is getting oxidized. But see, if it is cyanide or carbon monoxide, none of the complexes are getting oxidized, which means that if this inhibitor is rotenone, the electron transfer is not completely blocked. Whereas antimycin, it is a complex 3 inhibitor. Antimycin also, the electron transfer still take place to an extent. Okay, but if this is cyanide, sudden stopping of respiration, oxidation will not take place, oxygen consumption will not take place. So there is a sudden stopping of respiration, which means that in this question we have to choose. Rotenone is out of, it is not the answer. Dinitrophenol is not the answer. Oligomycin and antimycin are there. Then we have to choose oligomycin here, because if it's an oligomycin, we can see that oxygen consumption and respiration is stopped. But antimycin, as it is a complex 3 inhibitor, I will choose oligomycin as the answer. Antimycin is also an answer. I can't say that it is not the answer. But if cyanide was there, I would have chosen cyanide as the answer, not oligomycin. But if we are comparing antimycin versus oligomycin, I will choose always oligomycin. Antimycin versus cyanide, then I will choose cyanide. Cyanide versus oligomycin, I will choose cyanide, not oligomycin. But in this question, the best answer is oligomycin. I hope that is clear. So the next question is about the double strand break repair and the genes and the proteins involved. Which of the following is not involved in the repair of the DNA if there is a double strand break is a question. So the options are ATM, BRCA1, MSH2 and Q protein, Q70. So we will see which are the proteins involved in the double strand break repair. We know that there are two types of double strand break repair mechanism. One is the homologous recombination, the other is a non-homologous end joining. So if there is an homologous recombination, the proteins involved, number one is BRCA1 and 2, then NBS1, MRE11, RAD15, mRN complex that senses the DNA damage. So these are the proteins involved. Directly we are not seeing any ATM here, but I will tell what is the importance of ATM here later. So which means that not involved is the question. So we have seen BRCA1 is involved. Now you see the NHEJ, which is a double strand break repair mechanism. So for that, DNA ligase 4, DNA protein kinase, that is DNA PK, KU, that is Q protein 17, Q protein 86 and Artemis. This is a kind of endonuclease. So these are the proteins involved. For this NHEJ. So there you can see Q17 you can see. Now what about ATM? Now we are left with these two options, ATM and MSH2. So let me tell you what is the importance of ATM here. So ATM means ataxia telangiectasia mutated. This is one of the most critical regulator of every repair mechanism. Not only for double strand repair, for every DNA repair mechanism. So have you seen a complex here, MRN complex for HR. So what is that MRN complex? MRN is a complex of 3 proteins which are called as MR11, RAD50 and NBS1. That is called as MRN, right? So, in order to activate this MRN, what is required? This ATM. See, active ATM uses its kinase activity to activate this very important complex which is called as the MRN complex, which means that ATM is invariably required for this MRN complex that is required for double strand break repair mechanism, which means ATM is also involved here. Then what is not involved here is MSH2. So what is this MSH2 for? MSH2 is for a mechanism which is called as mismatch repair mechanism. There you can find MSH2 and MLH1. So let me tell you which are the other. I know this is very complicated, but still I will tell you which are the important proteins involved in other repair mechanism, where you can see XP. So whenever you see XP, you know that it is nucleotide excision repair. So XPACD and ERCC1. And MSH mismatch repair, I've already told you MSH2. And the next is base excision repair. Again, it is a long list. One is a glycosylase. Mute YH, if you remember the disorder, you can learn this mute YH from here. Then flap endonucleases, then XRCC1, DNA polymerase beta, DNA ligase 3, and a poly-ADP ribosylation protein which is called as PARP1. So these are the proteins. I know this is very complicated. At least learn this previous year question that here the answer is MSH2, and learn MSH2 is for mismatch repair. Anything which is related to XP, it is nucleotide excision repair. At least these 2 points, these few points you learn. So the answer here is MSH2. So another very complicated question that was asked in 2023 INICET was about sirtuins. So true about sirtuins are all except increase in lifespan, stimulated by nicotinamide, increased insulin sensitivity, support glucose metabolism. It has histone deacetylase activity and cause chromatin condensation. So I will just tell you about the sirtuins. Sirtuin is like a set of proteins which has something called as a common NAD plus binding domain. So their activity is, it has a deacylase activity, which means it is removing the acetyl or the acyl group, and an ADP ribosyl transferase. These are the two activities of this protein which is called as, or this enzyme which is called as sirtuin. Now, what are these sirtuin related to? These sirtuins are related to, they are implicated in aging. That is, it delays the aging process. Another is like, it is like if there is decreased sirtuin activity, then it causes cancer, it causes neurodegeneration, and it causes insulin resistance. Okay. Now, what about that stimulation and inhibition by nicotinamide? So for that I will tell you what is the role of this nicotinamide here. The activity of the sirtuin. So just see this picture. Don't learn this reaction. Just see this picture and understand what is happening there. So this is the activity of sirtuin, where you can see this is nicotinamide adenine dinucleotide, that is NAD+, and this is a compound which is an acetyl lysine. Now what you can see is this acetyl group is being transferred to this compound. At the same time, nicotinamide, which you can see here, so the nicotinamide, you can see this nicotinamide is being released from here, which means this nicotinamide is cut and released. At the same time, this acetyl group is added to this, which means that this is acetyl ADP ribose. Even if you don't learn that, that is fine. Now you just see this. By this reaction, what you can see is nicotinamide is a product. Now what is the action of product on the enzyme? The product usually inhibit the enzyme activity, right? So nicotinamide is not something which stimulates sirtuin activity. Nicotinamide is something which decreases the sirtuin activity. So that is the one of the option in that question. Now we will go back to that question and see the answer. True about Sirtuins are all except increase in lifespan. Yes, it is the correct answer. Stimulated by nicotinamide. No, it is a false statement here. Increase insulin sensitivity and support glucose metabolism, because it is going to increase the insulin sensitivity, and it has histone deacetylase activity, and it causes chromatin condensation. What do you mean by this deacetylase? We have seen an activity of Sirtuin, right? That is removal, that is adding of an acetyl group. But remember, it is a reversible reaction. So in one direction, you can see that acetyl group can be added. In other direction, you can see the acetyl group can be removed also. So it has acetyl transfer as well as deacetylase. Both activities are there. So it has histone deacetylase activity, and if it is deacetylation, definitely it causes chromatin condensation. So that is all answer. But stimulated by nicotinamide is not the answer, because it is inhibited by nicotinamide. So the answer here is B. So the next question was the question on the synthesis of testosterone from cholesterol. So this was the question which was asked. You know this question, but let me explain the synthesis of testosterone from cholesterol. So first and foremost, we know that cholesterol is a 27 carbon containing compound. So this cholesterol, the first reaction that happens is, from this 27, 6 carbons are being removed. So that is by an enzyme which is cutting between, or which is cutting a 6 carbon compound from that, and that enzyme is 2022 desmolase, because it is cutting the cholesterol between the 20 and the 22 carbon atom, where we can see that 6 carbon atom is being removed. So that's the first step for every hormone synthesis, for every steroid hormone synthesis, that is the 2022 desmolase, which cut the 6 carbon. Now once the 6 carbon is removed from 27, we know that there is a 21 carbon compound. Which are the steroid hormones with the 21 carbon atom? One is cortisol, the other is progesterone. Then there are certain steroid hormones with the 19 carbon atom, which is called as the androstenedione group, which has testosterone. Then another one is estradiol group, where you can see estradiol belongs to that group. Now the question is how cholesterol is being converted to testosterone. So whenever we are learning that, this is the reaction which is very, very complicated. But I will make it very simple for you. What you have to do is, you have to draw a line like this and 3 line like this. Right? Now in this line you write 17 alpha hydroxylase, and in the second line you write 1720 lyase, and in the third line you write 17 beta hydroxysteroid dehydrogenase, and this line is an enzyme which is called 3 beta hydroxysteroid dehydrogenase. So I will make it very simple for you. So this, once you have drawn this, what you have to keep is the pregnenolone. We have seen that pregnenolone, you have to keep it here, which is the mother of all steroid hormones. So once you keep the pregnenolone here, it can go in different way. Either it can go down like this. You can see here, it can go down, then here, so like this. So first enzyme here, second enzyme activity here. Now we can see the third enzyme activity here, then it crosses and it becomes testosterone. This is one way. Another way is, the pregnenolone which we have kept here goes in this way. Now then it crosses the 17 alpha hydroxylase, then no need to learn those compounds. Next it crosses the 1720 lyase, then a dehydrogenase, like this. So this is another way. So there are 2 pathways whereby this pregnenolone can be converted to testosterone. But in humans, which is the most common pathway? In humans, the most common pathway is this pathway, where it first 17 alpha, that is the 17 alpha hydroxylase, then the next is 1720 lyase, and the next is 17 beta hydroxysteroid dehydrogenase, and the next one is the 3 beta hydroxysteroid dehydrogenase. So this is the usual route. So the most common route is like this. This is what you have to understand. Now we will go back to this question and find the answer. The first in the order should be which enzyme? It should be 2022 desmolase, which means the B should be the answer. So we are left with the two options here, 1 and 3. Then what is the next enzyme? The next enzyme is the most common pathway we have seen. It is the 17-alpha hydroxylase. So that is the next enzyme. So A should be the second. Then we can see 1720 lyase. That should be the third one. And there is one more enzyme there. And after that you can see 3-beta hydroxysteroid dehydrogenase as well. So D is the next in the option. So the answer here is 3, that is BACD. So just to learn this one which I have taught you, that is you have to keep pregnenolone here and you have to reach testosterone. So for that you just draw one one vertical line and 3 horizontal line. One horizontal, the first line is 17-alpha hydroxylase. The second line is 1720 lyase, and the third horizontal line is the 17-beta hydroxysteroid dehydrogenase, and this is the 3-beta hydroxysteroid dehydrogenase. So we will cross this, and then we reach here, which means 17-alpha hydroxylase, 1720 lyase, then 17-beta hydroxysteroid dehydrogenase, and finally 3-beta hydroxysteroid dehydrogenase. So that is the correct order whereby cholesterol is being converted to testosterone. So that is also a very difficult question asked in one of the INICET exam. So the last question which I want to discuss is about a question on enzyme kinetics. So we know that this is an enzyme mediated reaction, and in this question it is given like the reaction mixture has 1000 times Km. What do you mean by this 1000 times Km? So we know this is the Km value. Now if we 1000 times Km is the substrate concentration, means it will be very far away, which means at 1000 times the Km in this reaction mixture, the kinetics that is followed is a zero order kinetics, which means if you double or triple or whatever increase in the substrate concentration, it is not going to change the velocity in any way. But you see the velocity against the enzyme concentration, it is directly proportional. So if you increase or decrease the enzyme concentration, it is definitely going to affect the VI. This is what happens in this reaction mixture. Now what is given here is a particular amount of substrate is converted to a particular amount of product in about 9 minutes. Now in this reaction mixture we are making some changes. First change is in the reaction mixture, the substrate concentration is like the enzyme concentration is reduced to 1/3 and the substrate concentration is doubled. So substrate concentration doubled means, is it going to increase the velocity in any way? No. Now enzyme concentration is 1/3 means the velocity should be also reduced to 1/3. When velocity is reduced to 1/3 means what will be the time taken? If a car is traveling from point A to point B at 100 km per hour, now if the velocity of the car is reduced to 50 km per hour, then it will take double the time, right? So like that, if the enzyme concentration is 1/3, it means that the time taken will be tripled, 3 times more. So initially if it is 9 minutes, then now it should be 9 into 3, and the answer here is 27 minutes. Okay. So these are some of the difficult questions asked in INICET, which I tried to explain it in a very short time. This is a shortest way that I could explain these tough questions. You know, the tough questions, some concepts has to be explained, which I've tried to explain in a very short time. And thank you for watching, and follow for more. Bye-bye.